Hello everyone, this is a very short video just on some DNA structure basics for A-level. Now DNA, as we know, is our genetic material, and in textbooks and on the internet, if I were to draw this particular shape here and throw in a few lines, people would recognise that this is the characteristic double helix shape of DNA. But what we're going to do is just in this video outline some of the basics about its structure and the nature of DNA. Let's start by saying what the letters DNA actually stand for. The D stands for deoxyribo. That's where the D comes from. The N comes from the word nucleic. And the A comes from the word acid. So DNA stands for deoxyribonucleic acid. It's because of the nature of the sugar that it contains. So let's think about what actually comprises of this DNA strand. So we've got here, if I would draw them as a few different shapes. So we've got here big blue ring. This is going to represent our phosphate. So we have phosphate within DNA. Just drawing different shapes just to visually make the different parts stand out. So we have what's called deoxyribose sugar. Not to be confused with RNA, which is ribonucleic acid. So this is a different kind of sugar. Then we have our four particular bases, which we're just going to roughly colour in here. So we'll have one red, we'll have one of them as blue, pink, and green. Is so I could draw these structures in a moment and it would make a little bit more sense. So in red we have A for adenine. So A stands for adenine. In blue we have T for thymine. In purple we have C for cytosine, and in green we have G for guanine, because a lot of people, when we think about DNA, are familiar with the letters A, T, C and G, but aren't really sure about what these four letters actually represent. So we'll put these up at the top here. A stands for adenine, and they're called the bases. They're nitrogenous bases. They contain nitrogen. The T is for thymine, the C, and at this level you would be expected to know what the letters represent. At key stage 4 you wouldn't. And the G represents, it was still worth knowing, represents guanine. Now they are four organic bases. The A and the G are double ring bases. So they have a double ring shape. So that's A and G. They have a double ring, and T and C are what we refer to as single ring bases. So they have a single ring structure. Now let's think about when we actually start to combine them. So if we were to have a deoxyribose sugar, so we'll just put that in place here. So let's say we've got a deoxyribose sugar there, and joined to that deoxyribose sugar we have our phosphate. I'm only very roughly sketching these in. And then we attach to that, let's say we attach to that organic base. So let's, for the sake of argument, just say we're going to add adenine. There. Then what we ultimately get is something called a mononucleotide. So this structure here, so the base with the sugar and the phosphate attached, we call that a mono, meaning single, nucleotide. So clearly if we had two of those together, if we were to continue this, in fact I'm going to shrink the screen down so that I can add a few more bits of information here. If we were to continue that and let's say put in another, I won't colour them in, another deoxyribose sugar with the phosphate and then put another base on, that would be a dinucleotide, di meaning two. So you can see that the more we add, the greater the structure, the size becomes. So we go from a mono to a di to a trinucleotide. Eventually that would be a polynucleotide strand. And that's what DNA ultimately is. If you were to take this DNA molecule that I've drawn here and unravel it, it would look a bit like a ladder structure almost. And I'm hoping that you can appreciate that this whole structure that I've drawn here is ultimately one section of this, if you like. It's that region there, for example. The backbone, the sides of this ladder of DNA are made of sugar and phosphate. So they provide a sort of sugar-phosphate backbone. That's this part here. And the rungs of this ladder, these red lines I've drawn, are these bases. The A's, the T, the C and the G. Now, via what's called complementary base pairing, you see that the A, the adenine base, would complementary pair with a T, a thymine. So if, for example, if I just split that, if I draw it slightly differently and have it as building blocks, if this were an adenine base, then this one would be a thymine, because adenine always goes with thymine. So on the diagram I've circled over here, this red was an A. That would base pair to, if I were to have something matching on the equivalent side, that would have to base pair to a thymine, and that would have been attached to, clearly, another sugar and a phosphate. So A and T go together, C and G go together. And that's how you form that sort of inner structure of our DNA molecules. So cytosine pairs with guanine. Now what you find is that they pair via hydrogen bonding. You get two hydrogen bonds forming between an A and a T, and you get three hydrogen bonds forming between a C and a G. Now that level of detail you'd only ever cover at A level. So just a few final things to think about when we look at this DNA structure. So we've outlined what it's made of, what it's mainly comprised of, the four bases, the phosphate, the sugar. We've described what a mononucleotide is and how they go together to form a polynucleotide. And we've said about hydrogen bonding between those bases. So I want to think a little bit about structure and function, because the structure of DNA makes it perfect for carrying out the job that it does, carrying our genetic information. For example, the double helix structure that I've drawn, those two intertwined sort of sugar-phosphate backbones, they make the DNA very stable. So the DNA doesn't break down or get damaged very easily. The DNA molecules generally are very long and are coiled up really tightly. So you can store quite a lot of genetic information in that small space. In fact, I think what we'll do is just make a small note of these, because these are really important points relating to structure function. So as I said, number one is the fact that it's a very stable molecule, so it doesn't get damaged. The second thing I said was the fact that it's coiled. So because it's coiled, you can essentially fit in a greater amount of genetic information. And DNA molecules have a paired structure, which make it easier to copy itself in a process known as semi-conservative replication. And that's important for cell division and passing information, genetic information, down from generation to generation. So having these A's and T's join and C's and G's, the nature of the molecule allows for DNA replication to occur, which I've talked about in another video. So the third important thing is that it has a sort of paired structure via these bases. I think one crucial thing to end off this video is some information about DNA storage. Because although the structure of DNA is the same in all organisms, eukaryotic, as in plant and animal cells, and prokaryotic cells, so there we're thinking about generally bacteria, store DNA in different ways. So we've outlined the structure, and on the left-hand side of this video I talked about the bases and forming the strands, and we've said about its structure and function, but I want to finish with storage of DNA, because in a eukaryotic cell we have linear DNA, and by linear DNA what I mean is the DNA exists as chromosomes, thread-like structures, each one made up of a long molecule of DNA. So we have, in fact I'm just going to corner off the page here to finish on this section by talking about storage. So in a eukaryotic cell in plants and animals, you have this DNA structure here. You've got these bases, rough sketch, you've got the bases between, but we coil it up into very, very small molecules called chromosomes. Now those chromosomes, and I've got a replicated pair of chromosomes there, that's actually what I've drawn. Those chromosomes, or that DNA if you like, is wound around something called histone proteins. So you actually have, it looks a bit like, if I were to just draw these three here, we'll draw the DNA strand in red. It looks a bit like that almost. This DNA strand is wrapped around what are called histones. Now they're proteins which help to support the DNA. Because it's going to be a very long linear strand, we need to give it some structural support. So those DNA molecules are wrapped around histone proteins and it's coiled up to form what we know as a single chromosome. And that happens in eukaryotic cells. So the DNA is wrapped around histone proteins to form a chromosome. Prokaryotic cells, so bacteria, have something a bit different. They have DNA as chromosomes, but the DNA molecules are shorter and actually circular. And it's not wrapped around proteins. So in a prokaryotic cell, you don't have these histone proteins. You have essentially circular chromosomes, if you like. So in a prokaryotic cell, you have DNA that floats freely in the cytoplasm. And in other videos, I've referred to it as a plasmid. So the DNA in a bacterial cell exists as a plasmid, a circular strand of DNA, free-floating, so not contained in a nucleus, that actually can fit into the cell by what's called supercoiling. So it condenses to become really, really small. It supercoils to fit into that cell. So whereas an animal and plant cell have what I've just described here in the box as a chromosome, prokaryotic cells don't have a nucleus, and so their DNA floats freely in the cytoplasm as short circular strands. And the big circular strand that we know of is called a plasmid. So that's a little bit about DNA storage. Okay, hope everything in this video helps. Thanks.